Hello, everybody. I'm Tareian Cazares. I'm a graduate student at the University of Cincinnati. I'm in the PhD program for immunology. My mentor is Dr. Emily Moraldi, who's a fantastic systems immunologist. And today I'll be presenting our work on maxATAC, a method for predicting transcription factor binding from ATAC-seq and DNA sequence. So as an immunology-focused lab, we're really interested in how immune cells behave in vivo, especially in disease contexts, with a specific focus on learning how genomic regulation contributes to different phenotypes. And our group models transcriptional regulatory networks to learn the genes and transcription factors that are important to these cell behavior. Our group uses transcriptional regulatory networks, which describe control of gene expression by transcription factors, and we use the method called the inferlator to model experimental gene expression data to describe these networks. Unfortunately, despite the utility of such a network, there are always challenges with the number of experimental samples that can be used to infer these networks. There are often more interactions and parameters to model than the much smaller sample sizes, which number in the dozens usually. But one way around this we found is to provide information about context-specific transcription factor binding in the cell type of interest. And this is done by either providing ChIP-seq, which is an experimental method for measuring TF binding, or using TF motif scanning, which is an alternative approach. So I just want to introduce transcription factors. In case you don't know, TF is a protein binding, DNA binding protein, which directly interprets the genome. It has two primary characteristics, one being that it binds a sequence, a specific sequence, and the second one being that it regulates transcription through some mechanism. Transcription factor binding is traditionally measured by ChIP-seq or other experimental methods where you use an antibody to pull down sequences bound by the TF, find it, and use next generation sequencing to map back to the reference genome and find regions of enrichment. But taking this approach is very costly and unfortunately not feasible right now because there are a lack of specific antibodies for all 1,600 transcription factors. And there's also a high cost of cell number and a high time for optimization of these types of experiments. For example, recently the largest collection of transcription factors experimental determination in any cell line was HepG2, where 208 transcription factors were assessed by the ENCODE consortia. But there are, alternatively, you could use experimental methods such as ATAC-seq, which measures chromatin accessibility, and use that information to predict the binding of transcription factors. ATAC-seq uses a TN5 transposase to probe DNA that is not bound by other proteins or TFs, and you find regions of accessible chromatin. Up in the top, the red track is just an example signal track of what that would look like. And in these regions of accessible chromatin, you would then use either a known motif that's been derived experimentally or some other information to predict the binding of a TF in that location if you find the sequence that matches enough. And we've been able to show that using an approach, which my mentor calls the poor man's massively ChIP-seq, which is ATAC-seq plus motif scanning, does drastically improve our performance in reconstructing these gene regulatory networks, especially in these immune cell types that are rare. However, when we compared the standard approach, which is motif scanning, for 60 TFs in one cell line, TM12878, which we had a lot of ChIP-seq data to compare to, we found that the actual recovery of experimental transcription factor binding sites was poor. And we used AUPR to measure or assess our results. And on average, our median was about 0.1, with the top transcription factor being CTCF, about 0.4, which has a well-defined binding site and is usually overlapping accessible chromatin regions. And this has also been noted by the community at large. So in 2017, the ENCODE Dream Challenge pioneered state-of-the-art methods for predicting transcription factor binding using DNA's hypersensitivity data. And they also used some other genomic features. And the methods that came out of this challenge were able to achieve a median AUPR of 0.4, so fourfold higher than what you can get with motif scanning alone. But these methods aren't widely used. And why is that? Well, the models don't exist. If you use motif scanning, you can predict about 1,200 TFs, even if it's not the best prediction. Whereas with these state-of-the-art models, you can get 12, and I think now 30. So our group developed maxATAC for transcription factor binding prediction from ATAC-seq. maxATAC uses DNA sequence and the single base pair resolved ATAC-seq signal to predict transcription factor binding in a new cell type. And this was a multi-lab effort that was cross-disciplinary. Our training data for the maxATAC model was derived from the ENCODE and GEO databases. We used ATAC-seq and ChIP-seq data to train our models. And we were able to develop 127 transcription factor models using these data set. I just wanted to touch on how we evaluated these models. For 74 models, we used what was called a round robin approach, where we held out one cell line and 2 chromosomes from the model training and validation process, so they've never been seen by our model. And then we use that third one for testing. And in this case, we had 3 cell lines, so we did 3 rounds, and then we choose the median AUPR to represent the TF model. And when we actually compare our performance to motif scanning, across the board, we do much better, a little bit closer on the lower end there of the AUPR spectrum, but much better overall. And I just wanted to highlight the TFs in red are TFs for which there are no characterized known motifs in any database. Our models learn the features from the ATAC-seq signal and the DNA sequence to predict those TFs. So I thought that was really awesome. And with the increasing popularity of single-cell ATAC-seq datasets and the proliferation of motif scanning methods for single-cell ATAC-seq, we wanted to benchmark our methods on predicting transcription factor binding and pseudobulk with single-cell ATAC-seq. So we used a publicly available dataset that mixed cell lines from 9 different cell lines, and we had 7 of those for which we had in our training dataset, and we were able to generate pseudobulk ATAC-seq signals from those populations and make predictions for transcription factors and compared that to motif scanning. And all except for one model performed much better with our maxATAC predictions. So next, we wanted to know whether our models could be used to predict TF bindings in primary cell lines. Since all of our training data was based in transformed cell lines, we really wanted to see if we can make those predictions in human cells, relevant populations like CD4 T cells, that had been isolated from humans and activated for 5 hours with anti-CD3 and anti-CD28. There was paired ChIP-seq for 3 transcription factors, so we can actually assess how we did with our predictions. And overall, we show compared to motif scanning, we do much better on recovery of genome-wide transcription factor binding sites and have approximately the same precision or recall at the higher confident predictions for compared to motif scanning. So the next part of my talk focuses on exploring how genetic variation exploring human genetic variation a little bit with maxATAC and identifying potential transcription factors implicated in disease susceptibility. And to do this, we used a wonderful data set from our collaborators that had from the Children's Hospital patients with atopic dermatitis versus healthy controls, where we had ATAC-seq, whole genome sequencing, and RNA-seq. And for these models, we used the donor-specific DNA sequence in addition to their ATAC-seq signal to make predictions at 2 disease risk loci that had been shown to have allele-dependent accessibility profiles. And so we used the RNA-seq data from these patients to identify 105 transcription factors that were minimally expressed in these CD4 T cells. And we made those predictions in one haplotype block that was surrounding the RTL1 gene and the TNRSF6B gene. And the TNFR gene is associated with heightened serum levels of the protein in patients with atopic dermatitis. The other loci we looked at was Lyme1, which is a transmembrane protein that controls effector T cell migration to sites of inflammation, and SLC2A4RG, which is a known E gene, which was found with EQTL analysis in activated CD4 T cells. So these 2 sites do have previous evidence for being involved in atopic dermatitis, and so we really wanted to see what could be the potential regulators that are binding in these regions or contributing to potential disease cause. And so we found 2 TFs, MIB and FOXP1, that had increased binding in atopic dermatitis patients compared to their age-matched controls. And we are currently in talks with our collaborators to follow up with ChIP-seq experiments in these same exact patient samples so that we can validate our predictions. So for more detailed information and a lot of the analysis that I did in our bioRxiv preprint, you can check out my poster or look on our bioRxiv revised manuscript. We have our user-friendly code base on GitHub, PyPy, and Docker Hub. All of the data that we use to train our models and evaluate is available on Zenodo, so you can get the raw TN5 signal, bigwigs, everything needed to develop your own method that's better than mine. And I want to thank my lab for all their help, our multiple collaborators at the Cincinnati Children's Hospital. Emily's actively recruiting postdocs and PhD students, so please look at our website for some cool projects. And follow me on Twitter. I'm looking for a new home soon. I should be graduating, so if you know anybody that needs a postdoc or research assistant, let me know. And follow me on Twitter at Tareian Cazares.